Welcome to the lecture cast on mucosal and regional immunology. Mucosa represents the membrane that lines the inner surface of the body. Like skin, it forms a barrier between the internal and the external environment. With an area of 400 square meter, the epithelium of adult mucosa represents the most frequent portal of entry for common infectious agents, antigens and carcinogens. Thus, highly efficient mucosal immunity is of paramount importance. Mucosal immune system is defined as a part of the immune system that responds and protects against the microbes that enters the body through the mucosal surfaces. The mucosal immune system is provided with a rich collection of lymphocytes and auxiliary immune cells like mast cells, IgE, IgA, and macrophages. These are present in the epithelia and the lamina propria of the mucosal surfaces. You can see in the picture the collection of lymphocytes in the intraepithelial and subepithelial lamina propria. These cells are large in number with a distinct phenotype and functional heterogeneity. Some of these cells are organized into a lymphoid follicle-like structure. They are called as mucosal associated lymphoid tissue. These are the sites of adaptive immune responses to the environmental antigens. The MALT tissue are classified into specialized aggregates like how we see it in Peyer's patches, or they can be as scattered isolated lymphoid follicles. The MALT tissue is distributed all throughout different mucosal surfaces of the body. When they are present in gut, they are called as GALT, gut associated lymphoid tissue. It comprises of Peyer's patches, appendix mesenteric lymph nodes and solitary lymph nodes. Those lymphoid tissue present in the nasopharyngeal area or the NALT tissue, the Walde's ring including the tonsils and the adenoids form the component of the NALT tissue. Other lymphoidal tissue associated with mucosa are BALT with the bronchial region, urogenital lymphoid tissue, and those lymphoid tissue associated with the exocrine glands like salivary, lacrimal, and mammary glands. The ultimate aim of mucosal immunity is to bring about the homeostasis, that is, the defense of the mucosal surfaces should be established in such a way that there is no impairment of the structural and the functional integrity of the mucosa. That is, the mucosal responses to the pathological stimuli should be limited to prevent the overstimulation of the immune system so that the integrity of the mucosa is preserved. Against a non-viable antigen, which is the potentially harmless or a self-antigen, a modest to no immune response is being elicited. Against a pathogenic mucosal antigen, a regulated immune response is elicited. What do you mean by regulated immune response? The regulated immune response is the heightening of the immune activity once the antigen is encountered. Following the elimination of the antigen, the immune system is brought back to normal sense. Thus, the hallmarks of the mucosal immunity includes the non-inflammatory effector mechanism by which the various immune mechanisms are mediated through a non-inflammatory process. There is immune tolerance against a self-antigen or a potentially a harmless antigen, and regulated immune response towards a pathogenic stimuli. The various mechanisms of mucosal immune system include non-specific mechanisms and specific mechanisms. Non-specific mechanisms include the physiological barriers as provided by the epithelial cells, cilia, mucus and other secretions, those given by the antimicrobial peptides, commensal bacteria and the pattern recognition receptors. Some of the specific mechanisms include those given by the antibody mediated, cell mediated and humoral mediated responses. Before going into the details of the immune mechanisms of mucosal immune system, let us see how an antigen is being presented to the mucosal surface. There are specialized M cells or the multifold cells which are present on top of the mucus associated lymphoid follicles. You have the galt and NALT, over which the MALT M cells they lie. These M cells, they are specialized epithelial cells. They do not have the microvilli. These M cells, they take the antigen from the lumen to be processed by the lymphoid follicle. In the follicle, there are presence of B cells which just on encountering these antigens, they get converted into specialized plasma cells. Those plasma cells, they produce the specific IgA or the immunoglobulin A to respond to the particular antigen. These are secreted through the lamina propria into the lumen. Thus, they counteract the specific antigen. We have the epithelial cells, for example, the enterocytes. These take up the soluble antigen with the help of MHC class 1 and class 2. They process and present them to the underlying T cells or the intraepithelial T cells for the immune tolerance or immune stimulation. There are dendritic cells which are present in the basal epithelial layer, or they can also be seen in the lamina propria, which protrude their pseudopores into the lumen to sample the antigen so that they activate and regulate the epithelial cells. Coming on to the mechanisms of mucosal immune system, there are non-specific barriers. Most predominantly, the epithelial cells play an important role. They have the intracellular junctions and the keratin formation, which prevents the transit of antigens and pathogens into the mucosal surface. Some of the epithelial cells, which are labeled as type 2 epithelial cells, they show stratification, that is, they constantly shed the superficial layer. Because of the superficial shedding, there is no colonization of bacteria happening in the epithelial layer. The mucus production by some epithelial cells, they act as a vital physiochemical barrier. The mucus produced along with the beating cilia, they form the mucociliary blanket. This mucociliary blanket, they effectively trap the antigen and expel the same. So in the various mucosal surfaces, you have secretions like tears, saliva and urine, which causes the washing actions. There are some of the reflexes like peristaltic movement, sneezing, coughing, which helps to expel the antigens. There are bactericidal compounds in some of the secretions. Example, an acid in a gastric juice, lactozyme in tears, saliva, lactoperoxidase in mammary secretions, and you have spermine and zinc in sperms. These help to kill the microorganisms encountered by the mucosa. The residual microbial flora, they have an extensive and vital interaction with the mucosa. They produce the microbial antagonism, that is, they suppress the growth of the various pathological bacteria and fungi by competing for the essential nutrients and also by producing various inhibitory substances. For an example, lactic acid produced by the vaginal flora help to get rid of the pathogenic fungi colonization. The antimicrobial peptides produced by mucosal epithelium, they forms a primary bacterial defense barrier. There are a group of antimicrobial peptides called defensins. They get inserted themselves into the bacterial membrane and causes the disruption of the same, because of which there is a loss of cellular contents, ultimately causing the lysis of the bacteria. Similar mechanism is being shown by salivary antimicrobial compounds like lysozyme, histatin, beta defensin and human cathelicidin, which also affects the cell wall integrity and pore formation. Some of them also trigger the immune mechanisms, ultimately causing the elimination of the pathogen. Some of the phagocytes and the epithelial cells, they express the innate pattern recognition receptors. These receptors, they recognize the microbial associated molecular patterns. These patterns are nothing but lipopolysaccharide in case of a gram-negative bacteria, or a lipoteichoic acid in case of a gram-positive bacteria, and a glycolipid in case of the mycobacteria. Or when the innate pattern recognition receptors, they combined with the microbial associated molecular patterns, also called as PAMP, there is an induction of diverse amounts of cytokines, which causes influx, activation and differentiation of myeloid and lymphoid cells. The other mechanism, as soon as a PRR encounters the PAMP, is that there is initiation of phagocytosis. That is, after the PRR combines with the PAMP, there is NF kappa B pathway which gets activated, because of which there is stimulation of phagocytosis. Thus, the phagocytes, they throw their pseudopods, engulf the antigen, and they form the phagosome. And as soon as the antigen is internalized inside, there is combination with the cytoplasmic granules of the phagocytes, ultimately causing various microbicidal mechanisms like oxygenated and nitrogen mediated pathways. Soon, a soluble debris is being expelled after the lysis of the microorganism. Various specific mechanisms are provided by cell mediated, humoral mediated, and antibody mediated. The specific mechanisms of the mucosal immune system are provided by the lymphocytes and antibodies. IgA is the dominant type of antibody which is seen associated with the mucosal surface. More than systemic circulation, it is commonly seen and associated with the mucosa. The appropriate site where the adaptive response or a specific response of the mucosal immunity occurs are the MALT or the mucosal associated lymphoid tissue. These MALT tissue, they have follicle. The center of the follicle is B cell rich, also called as a germinal center. These are the sites where the antibodies which are specific to an antigen are being produced. In the interfollicular region, you have the CD positive T lymphocyte, and these are the sites of cell mediated immune responses. Now we'll see the role of IgA in mucosal immunity. These IgA antibodies are seen bathing in the extracellular body fluids, tears, saliva, nasal secretions, and also the fluids, intestine and the lung. These are polymeric. They have 4 to 8 antigen binding sites, thus they have the enhanced avidity or attraction to the antigen. These coat the bacteria and the virus and various other microorganisms. They prevent the adherence of them to the mucosal membrane. These IgA, they attach to the Fc receptor in the macrophages and the neutrophils, mediating phagocytosis. An IgA molecule is a dimer which has been cross-linked with the help of a J chain. These are produced in the lamina propria. They are being transported with the help of a poly-Ig receptor on the cell surface of an epithelial cell. These combined with the IgA which is produced and transport them through the membrane, and finally after the lysis proteolytic cleavage of the transporting molecule, the secreted IgA occurs in the luminal surface. If the microorganisms succeed penetrating the IgA barrier, the IgE next come into play. They are the next line of defense. The complement derived anaphylotoxins and the IgA, they utilize the mast cells. They interact with the mast cells to cause the release of pre-secreted mediators of inflammation, which causes the local amplification of immune responses. The IgA, when it contacts with the antigen, it binds to the Fc receptor of the mast cells. Due to this, there is a release of mediators. The released histamine, they dilate the vasculature, causing transudation of IgE and complement into the area. These They opsonize the organism, causing the phagocytosis. The sum of the chemo factors which has also been released from the mast cells, they attract the neutrophils, eosinophils and other effector cells, which causes the phagocytosis. If the organism is too large to be engulfed, antibody dependent cellular toxicity, which is an extracellular mechanism, they mediate killing of the organism. Finally the organism is disposed. Now let's see about the role of T lymphocytes in mucosal immunity. The variety of T lymphocytes are contained in the mucosa. They are seen in the intraepithelial, subepithelial lamina propria and also associated with MALT tissue. The intraepithelial T cells are predominantly CD8 positive lymphocytes and also memory T cells. The CD8 positive T cells or the cytotoxic cells, they cause the innate and adaptive cytotoxic mechanism. The memory T cells, they rapidly get reactivated to provide for the immediate cytotoxic responses needed for an infection. The lymphocytes are also present in the lamina propria. They are CD positive T helper cells. These T helper cells, they recognize and respond to the antigens in the lymph nodes, draining the particular mucosal surface, and they migrate back to the mucosa to populate the lamina propria. These are predominantly Th2 type of lymphocytes. They activate B cells and antibody production. Some of them are only Th1 type of cells, that is they are in association with Mh2, they stimulate the cell mediated immune response. Whereas some of the helper cells are Th17 type due to the stimulation of IL-17 and IL-22, they cause the secretion of innate antimicrobial peptide and also they recruit the neutrophils through chemotaxis. Reactivity versus tolerance. How does a mucosal immune system distinguish between a self and a non-self antigen? They have to differentiate between the commensal microbiota, an essentially harmless food antigen, from a noxious pathogenic stimuli. How does a mucosal immune system distinguish between a self and a non-self antigen? They do this with the help of major histocompatibility antigen and also the co-stimulation. That is, when a particular cell recognizes the pathogen associated molecular pattern, the MHC molecule co-stimulate, they get upregulated and there is also co-stimulatory molecular expression. Only after seeing the co-stimulation and molecular MHC upregulation, there is activation of immune response. When there is absence of the same, you have immune tolerance. Replicating rather than an inactive microbe more likely induce immune response. There are various cytokines and mediators which have been produced by accessory immune cells which primes the microenvironment for a particular adaptive response. For example, if there is interleukin 2, 12, interferon gamma which is being produced, Th1 type of cell mediated immunity gets activated. If there is interleukin 4, 6, 5 and 10 being produced, Th2 type of cell mediated immune response being activated. The interleukin 12 and interferon gamma, they break the tolerance state and reactivate the immune stimulation or heightened response. There are certain suppressor cytokines like interleukin 10 and TGF beta which causes the immune suppression or tolerance. The FOXP3 is a gene which converts the naive T cell into a T regulatory cell in presence of TGF beta. These regulatory cells present in the mucosa, they dampen or suppress the reactive antigen specific cells. There is a particular contributory role which is shown by the intestinal microflora or mucosal microflora. Some of the organisms, they release the short fatty acid, which also contributes for the conversion of naive T cell to a T regulatory cell, ultimately leading to immune tolerance. The mucosal immune system acts as an integrated network. When an antigen is being exposed to one site, particular site of the mucosa, there is production of specific antibody at the other mucosal or the secretory sites also. How is it? There's a generalized immune responses are brought about by means of communication networks. The concept of common mucosal immune system also extends to the exocrine glands like the lacrimal, salivary and genital tract glands also. These are brought about with the help of dissemination of cells from the inducer site to the effector site. This is a particular novel immunization strategy which has been much exploited with mucosal administered vaccines. Most of the current vaccines which are being administered by systemic route do not adequately elicit the protective response from the mucosa. The mucosal route of delivery is being utilized to produce a secretory antibody and memory effector T cells for the desired responses. The mucosal immunization reduces the carriage of pathogen at the mucosal surface, thus they reduce the incidence of transfer of organisms in a communicable disease. This particular strategy is also being used by the principle of herd immunity. The exploitation of mucosal immune system has received immense attention in recent years. The attractive properties and advantages of mucosal vaccines has resulted in establishment of several commercial institutions. Although the anticipated targeted vaccines have not got the success in the clinical trials, much of a success, various mucosal adjuvants are the various vehicles for the transport of mucosal immunity has been tried. The efforts have been given to exploit the mucosal tolerance for the clinical benefit. Novel approaches like immune regulatory cytokines, delivery systems, and adjuvants are pursued. The many mucosal diseases like asthma and Crohn's disease are on rise. Understanding the homeostasis in the mucosal system holds key to find the preventive strategies for these common diseases. These are the references for further study. Thank you.